worked with sensory systems, hearing and smelling and also seeing, and the methods they are behind it.
Eyes are very complex. If you look at seeing, at vision itself, it's a real complex story. Every animal has its own vision. Not every animal sees the same. Some see colors, some do not.
It begins in photoreceptor cell, and it's the best, I think, the best cell known in science with this whole signal transduction cascade. The inner segment where the nucleus is also, and the whole production of the proteins, they have to move to the outer segments where this signal transduction takes place. And the only intracellular connection between these parts or compartments of the cell is connecting cilium. We are working on a protein which is discovered first in a disease. It's a polydactyly disease and involved in dwarfism. It's a dwarfism disease also, and we know that this protein is involved in mitosis, in cell division, but also in it's located at connecting or at cilia itself.
We just have to know where this protein interacts with other proteins. Imaging is very important in different parts of our lab, especially on protein level for Western blotting or protein geles is very important for us because we are interested in our proteins in the retina, especially on different stages of development of the mouse, and we have to quantify them and to measure different proteins in the same extract, in the same experiments. Is the protein there? Is the splice variant there? How much is it expressed or is it not?
One of the problems we have is to quantificate proteins on perhaps different stage of development of the retina. I like about this new ChemiDoc MP system that we have these multiplex features with several colors, about up to 3 colors. We can quantificate it quite easier because we can use several antibodies, up to 3 antibodies, one for the protein of interest and the other 2 perhaps for quantification protein, which is expressed in the whole retina.
So the real good thing on this system is I can take my one blot I got out of this one animal and I can show all the proteins I'm interested in one lane. In the field of vision, we're working with a lot of animals and we have to think about a lot of different kinds of vision, going from the very basic vision of plain white light to this bright color vision a monkey or a human being can do. And so I guess it's a little bit alike with these instruments. Just measuring black and white, you just can measure one color. And now this new ChemiDoc MP evolved this kind of color vision. This imaging possibility to image these different colors is anyway a great step in evolution.
So we think that the ChemiDoc MP system would allow us more or deeper look inside the expression patterns of proteins, perhaps in developing retina, to get more look inside in these human diseases.